All right, let's just jump right in. Welcome to this explainer. Today we are taking a highly structured journey through humanity's biological toolkit. We're going to look at exactly how we intervene in disease, scaling all the way from single molecules right up to entire complex organisms.
So here's our roadmap for today. Think of this as your high-yield crash course in modern biological interventions. We're covering 1. Organization and disease basics, 2. Immunology, the vaccine revolution, 3. Oncology, chemo versus radiation, 4. Gene therapy and vectors, 5. Reproductive cloning and SCNT, and finally 6. 10-second exam summary.
Okay, Section 1. Organization and disease basics. The fundamental levels of life.
Before we can even think about fixing a biological problem, we really need to understand exactly where that problem lives. Let's establish the physical framework of life. Biological organization follows this perfect logical flow. It starts from subatomic particles, down at the bottom, building up to atoms, molecules, organelles, cells, tissues, organs, and eventually the entire organism and the biosphere. Now, why does this actually matter? Well, it's simple. Any disease is essentially just a deviation in normal structure or function at one of these specific organizational levels. So to intervene successfully, we've got to target the exact right level.
And getting down to that foundational subatomic and atomic level, we are essentially just walking chemistry sets. The big three bioelements absolutely dominate our structure. Oxygen makes up a massive 65% of our body mass, followed by carbon at 18% and hydrogen at 10%. Biological life relies heavily on these specific elements, which is a really great reminder that biology is, ultimately, just highly localized chemical reactions.
Moving on to Section 2. 2. Immunology, the vaccine revolution. Preventative biological tools.
Let's move up the biological hierarchy to our first major tool, immunology, and specifically how we use preventative measures. The historical pathway of preventative medicine actually kicks off back in 1796. The father of vaccination, Edward Jenner, noticed something fascinating. Milkmaids who caught a really mild disease called cowpox didn't end up getting smallpox. So he actually took blisters from a milkmaid and inoculated a 9-year-old boy, effectively proving the whole concept of immunity. Fast forward to 1967, the World Health Organization launched an intensified eradication plan, and by 1980 they declared smallpox completely eradicated. To this day, it remains the only infectious disease in human history to achieve that distinction. Pretty amazing, right?
So how exactly do we train our immune system's memory cells without, actually causing the disease? Well, one of our primary tools is the attenuated vaccine. By definition, this is created by reducing the virulence, meaning the harmful effects, of a pathogen. It is fundamentally an alive vaccine, but the attenuation process makes it completely harmless.
Now, if you are prepping for an exam right now, consider this a giant flashing red warning light. Do not fall into the really common exam trap of confusing your vaccine types. Attenuated vaccines are live, but weakened. They are entirely distinct from inactivated, killed whole vaccines. You'll definitely want to memorize that distinction.
All right, Section 3. Oncology. Chemo versus radiation. Reactive interventions for cancer.
So we're shifting away from preventative tools now to focus on reactive interventions. Specifically, we're looking at oncology and cancer, which at its core is a disease defined by false mitosis and abnormal cell division. Let's look at our two main approaches. First, chemotherapy. Think of our analogy anchor here. Chemo is like a systemic carpet bombing. It uses powerful cytotoxic drugs given intravenously or orally to just kill fast-growing cells all throughout the entire body. Then we have radiation therapy. Think of this as our localized sniper. It uses high-energy beams like X-rays to target and kill cancer cells at one highly specific tumor site. Because chemo is systemic, its side effects are felt throughout the whole body, whereas radiation side effects are typically limited right to the treatment area.
But the absolute crucial point here is that these interventions are not mutually exclusive. Your oncology team might use concurrent therapy, meaning they give both at the exact same time. The chemotherapy can actually make the cancer cells more sensitive to the localized radiation, targeting the tumor directly. They work together.
Next up, Section 4. Gene therapy and vectors. Cellular modification tools.
This brings us back down to the microscopic level. What if we don't just want to kill abnormal cells, but we actually want to fix the faulty genetics themselves? Enter gene therapy. But we absolutely have to make a very sharp distinction here between two types. Somatic gene therapy targets your regular, undifferentiated body cells, meaning any genetic fix stops with you. Germline gene therapy, however, inserts genes into embryonic or germ cells, and this means the genetic alteration is permanently passed down to future offspring. As you can probably guess, germline therapy is surrounded by intense, heated ethical controversies because it literally alters the human lineage.
Okay, but to actually get these therapeutic genes into the cells, we need delivery trucks, or you can think of them as cellular Trojan horses. We use viral vectors for this. Adenoviruses are non-enveloped with double-stranded DNA, and they can infect a pretty wide variety of cell types. Then you have adeno-associated viruses, or AAVs, which are much smaller, have single-stranded DNA, and are naturally replication defective. They actually require a helper virus, like herpes or some form of genotoxic stress, just to be able to replicate inside a host cell.
So, how do scientists actually harvest these vectors in a lab? Let's quickly step through this chronological milestone sequence. Step 1. Scientists use bacteria as little microscopic factories to replicate the altered viral vectors. Step 2. They apply a detergent, which ruptures the bacterial cell walls and frees the DNA. Step 3. A purifier is used to physically separate the larger bacterial DNA from the smaller vector DNA loop. And finally, step 4, the syringe is filled with just the purified vectors, ready to deliver that genetic medicine right into the patient.
Section 5. 5. Reproductive cloning and SCNT, Advanced Reproductive Biotechnology.
We are escalating to the absolute peak of our biological toolkit now, the replication of entire complex organisms. The rapid evolution of cloning actually started way back in 1952 with the first cloned frogs by scientist Briggs and King. But the true revolution that shocked the globe hit in 1996 with Dolly the sheep at the Roslin Institute in Scotland. And it definitely didn't stop there. By 2005, South Korean researchers successfully brought us Snuppy, the very first cloned dog.
Now, the specific methodology they used to create Dolly is a highly testable concept called somatic cell nuclear transfer, or SCNT. This involves taking the nucleus from an adult somatic cell, in Dolly's case it was an udder cell, and inserting it directly into an egg cell that has had its own nucleus removed. We call that an enucleated egg cell.
It took 277 manipulated eggs to achieve just one single successful birth of Dolly, and when they cloned Snoopy the dog, it took over 1,000 cloned embryos implanted into 123 surrogate wombs to get just 3 successful pregnancies. This really illustrates the immense, staggering difficulty and the massive failure rate of SCNT.
But why go through all that effort, right? Well, aside from research, there are real-world transgenic applications. For example, in Argentina, cows were cloned specifically to produce milk that actually contains human proteins. This is absolutely incredible science for sure, but it instantly sparks some really fierce ethical debates regarding food safety and the limits of manipulating animal genetics for human benefit.
Finally, Section 6. 6. 10-second exam summary. The high-yield corner.
All right, if you've been taking notes, put your pen down for just a second. Welcome to the High Yield Corner for our ultimate rapid-fire exam revision session. Here are your top 4 absolute must-know takeaways. 1. Attenuated vaccines are live but weakened. 2. Chemotherapy is a systemic carpet bomb, while radiation is a localized sniper. 3. Germline gene therapy affects embryonic cells and is permanently inherited by offspring. And 4. SCNT cloning requires a somatic nucleus placed into an enucleated egg. Got it?
We've covered a massive amount of ground today, looking at how humans manipulate biology to cure, prevent, and replicate. But I want to leave you with this final, somewhat provocative thought. As our toolkit inevitably evolves from Jenner scratching cowpox into a 9-year-old boy's arm, all the way up to somatic cell nuclear transfer and transgenic mammals, where exactly do we draw the ethical line for human applications? The science is already here. It's the boundaries we still have yet to figure out. Thanks so much for joining me on this explainer, and keep learning.